Hello again, welcome back. So today what we want to tackle is talking about animals and their diversity. This is one of my favorite lectures. I love animals. I've studied animals for I guess over 20 years now, and that's where most of my research is concentrated in. I do a lot of research on reptiles and amphibians, so I do like this particular topic. So I want you to think to yourself for a second, what is an animal? How is that different than a plant? How are they different than fungi? So an animal has these characteristics. They're going to be multicellular, so they're made of more than one cell. They are going to be heterotrophic, which means they have to eat or ingest food and digest that food that they eat. Their cells lack cell walls. This is kind of a unique characteristic. You know, fungi have cell walls, protists have cell walls, plants have cell walls, bacteria and archaeans have cell walls, but animals do not. They do make a protein called collagen, and this is the most abundant protein that seems to be in the animal body. And this protein has quaternary structure, which means it's made of more than one protein put together. It's actually 3 twisted strands, and this seems to be a really strong building material. Animals do reproduce sexually, so the sperm cell and the egg cell will unite to form a zygote. The zygote will undergo, cell division to create various stages of little cellular spheres. When you get to the blastosphere, it has a solid outside, and then it has a hollow core. And this will actually go through, this blastula will go through gastrulation, where you'll form 3 layers of tissues, and those tissues will then form, more complex structures. Some go on to form fetuses and other structures, and we'll talk about that perhaps later. So most of these things we call animals are going to contain complex tissues. They're going to contain connective tissues, muscle tissue, epithelial tissue, nervous tissue. But oddly enough, some don't contain tissues or organs. So that's not necessarily a characteristic of an animal containing organs. I guess most of you would have thought that, animals would have had brains, but not all animals have brains. So they do contain Hox genes. Hox genes control the development of specific animal form, and this is unique to animals. I'll show you what those Hox genes do in just one second. So here we see a fly, and you can see that it's color-coded. And what this is trying to show you is that there are specific genes that code for different sections of this particular fly's body. So if you notice, these particular areas are contained by these master control genes that we call Hox genes. Now if you mutate the Hox gene, you can mutate the body part. Okay, so this is the adult form up here at the top. This is the larval form down here at the bottom. But as you notice, the larval form, there are certain coded instructions that code for particular segments of the creature's body. And so when you think about how does an insect have 3 body sections, it's because of Hox genes. And if these Hox genes are mutated, you get, mutated body parts. Humans also have Hox genes. So if you notice, these particular genes right here will code for this part of the embryo, so which eventually leads to this part of the adult form. And these particular Hox genes code for these particular segments, or if you notice over here on the adult form, it forms those particular sections. So these are very complex master control genes, and you can mutate these genes very easily to get new forms, new life forms. So if you think about the evolution of animals, how do we get so many different kinds of animals? Mutation of the Hox genes probably led to that particular phenomenon. So we can see in this picture here the normal fly in the middle, and you can see an antenna. Let me change the color here so you can see the antenna. So here are the antennae. And if you take and mutate a Hox gene, you can actually cause a leg to form where there should be an antenna. Okay, so they have manipulated these things and figured this stuff out pretty well. So what happens when Hox genes are mutated? Well, you can get extra toes, you can get body parts hanging out of you, extra body parts. So this is not uncommon. You know, you can go to the internet and type in, you know, Hox genes in humans and see all kinds of images of people growing extra body parts. Okay, so I want to talk about animal phylogeny. Now, if you remember, phylogeny is the evolutionary history of a creature. So we want to talk about how these creatures are, um, you know, which ones are more simplistic, which ones are more complex, and kind of how they evolved over time. So the common ancestor of all animals is probably some kind of protist organism. I'll show you why that we think that in just a second, but it was probably a protist. And if you notice in this particular chart here, there are two main groupings. We have Parazoa and we have Metazoa. Parazoa are animals that don't have tissues, organs, or systems. We still consider them to be animals because they have the characteristics that animals have in common. Metazoa do have true tissues. Some don't have organs, but others do have organs and systems. So we'll begin with Parazoa today. This is a cladogram showing you all animals. And then you can see that animals branched off into Parazoa and Metazoa. Parazoa have no true tissues, and the phylum Porifera or sponges will be an example of that particular group. Eumetazoans do have specialized tissues, and they're going to basically be differentiated between each other by symmetry and by the number of layers of cells they start out with. So if we look at Radiata, Radiata has radial symmetry, which means you can take and cut the organism like a pizza into many different halves and get equal halves. They also only have two cell layers that they start out with. They're called diploblastic. And some members of those include the ctenophores and cnidarians, or jellyfish and comb jellies. Bilateria have bilateral symmetry, which means you can take and cut them in one way to get equal halves. They also are tripoblastic, or made of 3 layers of cells when they begin. And then many of them have a body cavity. This chart doesn't really truly reflect what's reality, but we'll just go with it for now. There are some with no coelom or body cavity. Others of them do possess a body cavity. There are then other classifications or differentiations, protostomia and deuterostomia. This means, you know, the first pore that forms in the embryo, if it forms a mouth, it's a protostome. If it forms an anus, it's a deuterostome. So we'll go over those characteristics. Molecularly, we can differentiate Echetozoa from Leucotrophozoa, and we'll talk about each of these particular groups. Okay, so that's a cladogram of the kind of a modern cladogram of animals. As I indicated before, we believe that protists were the ancestors of animals because you can actually find protists alive today, like this choanoflagellate protist. It's an animal-like protist, and it shares very, very strong commonalities between cells that are found inside of sponges. Sponges seem to be the most simplistic animal, and they do have characteristics very similar to protists. So I'm going to say this one last time, but the protist seems to be where animals came from in a phylogically, looking at it from an evolutionary standpoint. This is where they came from. Why? Because they share common characteristics and they look very similar to what you can see in modern day sponges. Okay, so let's talk about Parazoa. You might see that that's a weird word, but it means beside animal. Para means beside animal. And the phylum Porifera is the grouping of creatures that are in this particular major group. These particular animals lack true tissues. The definition of a tissue is where you have cells that work together to perform a particular function, and they just don't have that. They seem to be a collection of cells that will work together, but not truly like epithelial tissue or muscle tissue or nerve tissue. They are beautiful creatures. They come in many different colors and many different forms. They do not have organs or systems, so they don't have a nervous system or a muscular system. They do not actually have symmetry, so they're asymmetrical. That means you can't cut them any way and get equal halves. They are sessile, which means they live on the bottom of the water or in fresh water or the salt water, but they do have free-living larvae. So the larvae do have little cilia and can swim around, move with water currents, and land in new locations. They do get their food by filtering little microscopic organisms out of water, so they're filter feeders. This makes them ecologically important because they do clean water up and control plankton populations or algae populations. They can reproduce in two ways. They do have sexual reproduction with sperm cells and egg cells, and they can reproduce asexually by forming little buds. This is where a little portion of their body, so if I just drew a sponge here, a little portion of the body breaks off and it forms a new sponge. They can do fragmentation where they form, you just cut it into pieces and each piece becomes a new sponge, and they produce these little resistant structures called gemmules, which can resist even drying out, and when they're put back in water they will sprout back out into a sponge. It's almost really hard to kill these things. You can put them in a blender, blend them up, and then it'll actually reattach itself and form a new sponge. So very resistant organisms. Sometimes, when I say simplistic, I don't mean that in a derogatory term. These are simplistic compared to other animals, but they're very successful in their simplicity. So if we look at the life cycle of a sponge, here's a little bud. So this would be an asexual form of reproduction where they form a new sponge from a bud. They do have the ability to produce and release sperm cells, and this is showing inside the sponge are where the eggs are. The sperm cells will be taken up by what we call a collar cell and brought to the egg cell so it can fertilize it. So if fertilization does occur, there will be a little embryo that will form. That little embryo will become planktonic, which means it'll float around in the ocean, be released to float around the ocean. And it has little cilia, little flagella that help it to move around. Flagella in this particular example, not cilia. And once it settles to the bottom, away from the parent, it can grow into a new sponge. This is just a little cladogram showing you modern-day sponges and how we think that they're kind of organized into groupings. So one of the early characteristics is an internal system of pores and canals for water flow. Water does flow into these creatures and then out of these creatures, as I'll show you in just a little bit. But if they have these little parts inside their bodies called spicules made of calcium carbonate, we put them in the group Calcarea. If they have siliceous spicules, which means spicules made out of silica or like a sand-like material, they can be put into these other groups. So the Demospongiae would be the types of sponges that have siliceous spicules, but they're not in these really interesting six-sided organization. So if they are six-sided, we put them into hexaactinidia, and so that's the three main groups or classes of sponges. Remember, you're in the kingdom Animalia, phylum Porifera, and then here's the three classes of sponges. Make sure you follow your study guide so you understand how much of that you need to know for testing purposes. Alright, so let's take and we'll look at this organization. So generally speaking, water comes in through these little pores and it exits the osculum at the top of this particular diagram right here. So we have all different kinds of areas where water can come in. Water comes in to filter, and water exits to remove waste products and also to take out sperm cells. If you notice in the more advanced drawing over here, the more I guess complex drawing over here, you can see its organization. So it takes the body wall and it shows you how it's organized. So we'll just go through and look at each of the different parts. We'll start with the choanocyte. The choanocyte generates a water current and it filters food particles from the water. So there is a flagellum. It has a flagellum, and then it has this little collar of almost like a membrane that's divided into parts, and that flagellum will circulate around in a circular motion, and then particles will get trapped on these little parts right here and then be brought into the choanocyte. The choanocyte will then take and move the food particles to these cells called amoebocytes. Amoebocytes are going to deliver nutrients to cells, so it'll move around and deliver nutrients. It can also differentiate into other cell types if those other cell types are needed. So a sclerocyte, it will be the part that secretes and makes the material that is made into spicules. Spicules can be sharp, they can add structure or add protection to the sponge so other things won't come around and actually eat it. So they're very sharp and very pointy and give structural support to the sponge. A porocyte is a cell that controls the flow of water through the sponge, so they create little water canals. You can also have egg cells, and you can have pinacocytes. Pinacocytes form the outer covering of the sponge, and these may also even phagocytose or eat food particles. We then have a chelanocyte or a lophocyte, and these secrete collagen, which is a structural building material that gives structural support to the actual sponge. And that's about it. You can study the sponge pretty quickly and learn all of its parts. Notice there's no systems here. There's really no like muscle tissue or nervous tissue or things of this nature. And so it's more simplistic compared to some of the animals we'll study in the future. Sponges lack complex nervous, muscular, circulatory, digestive, reproductive, and excretory systems. They're going to, instead of having a circulatory system and a respiratory system, and an excretory system, their circulation, gas exchange, and excretion is done by diffusion of water. Water comes in, it brings gases in, water goes out, taking waste products with it. So all of that's done just through the diffusion of water into and out of the sponge. So digestion is accomplished by the choanocytes and the amoebocytes. So these are two parts that will do that. So instead of having a mouth, esophagus, stomach, small intestine, large intestine, they just have cells that undergo digestion. And this is just showing you how they do derive their food. Here's your flagellum right here. This will basically go around in a circular fashion, create a water current that brings food. These are little extensions of the cell membrane, and it forms almost like a collar. So around the flagellum that you can see sticking out here. So the water comes in, little food particles they have here in this little brown color. They'll get trapped in mucus that will be covering the surface of these extensions of the cell membrane, and the food will be brought into down and into the cell. It creates little food vacuoles. The little food vacuoles are transported and they move over to the amoebocyte. The amoebocyte will then transfer the food to different areas of the body. This is just a little video. I'm not going to show all of it, just showing how the food comes in. So this is a huge sponge. Fish have a hard time eating that because of the spicules, although they will pick at it to see if they can. This person's injecting dye here, a green dye that you can see. And so you'll see that the sponge will bring that dye through, and the dye will exit through the osculum at the top. So it's going through porocytes, and you can see they're putting the camera over there. You can see it's gone through, and now it's exiting through the osculum. So that's pretty cool. The full-length video, if you just want to keep looking at that, I'll put on Blackboard for you to look at. Alright, so we'll move on to the more complex animals, the eumetazoans. Let me go back. So the eumetazoans. And so you can see that we can divide those into two major groups based on several different characteristics. We can base it based on radial or bilateral symmetry. So radiata means that you have radial symmetry. So you can cut it in many different ways to get equal halves. Bilateria means you can only cut it one way to get equal halves. So that's called bilateral symmetry. If we notice that radiata has two germ cell layers, so they have two layers of cells when they first begin, and that will form the whole organism, whereas Bilateria is going to have three layers of cells. So if you have two layers of cells, you're called diploblastic, or you're a diploblast. If you have three layers of cells, you are known as a triploblast. So if you look at the diploblast, you have two layers of cells. The outer layer is called ectoderm, the inner layer is called endoderm, and then there's a non-living kind of middle layer that is in between the two layers. If you're a triploblast, you have not only endoderm, the inner layer, you have the outer layer, but you also have a middle layer called mesoderm. Now, we'll go through in just a second and show you what these become, but each of these layers of tissues become certain things for the animal. So if you are triploblastic, have three layers of cells, the endoderm or inner lining is going to become things like lung cells, thyroid cells, digestive tract, those particular parts of your body. And yes, you had endoderm at one time that became all the inner linings of your body. The ectoderm becomes all the outer linings of your body. It becomes skin cells, pigment cells in your skin, and it becomes your brain and spinal cord. The mesoderm, or the middle layer, becomes all of the muscle tissue, cardiac muscle tissue, skeletal muscle tissue, smooth muscle tissue. So we have three layers of muscle tissue. It'll become your blood cells. It'll become your kidneys. So it becomes many, many different kinds of parts. Okay, let's go and we'll talk about radiata right now. Radiata is going to be the radially symmetrical animals. So yes, one of their characteristics is radial symmetry. Another major characteristic is that its body plan includes two germ cell layers. So it actually has endoderm and it has ectoderm. So the ectoderm will become epidermis, an outer skin-like layer, and the endoderm will become the gastrodermis, or the inner lining of the digestive cavity. These particular organisms do have a gastrovascular cavity. It has a mouth anus. The mouth anus serves as the mouth and the anus. So food comes in through the mouth. It goes out through the same exit hole, the anus, and food is digested in this cavity that we call the gastrovascular cavity. So digestive enzymes are dumped onto the food, and the food is digested, and whatever is not digested is excreted through the mouth anus and excreted out. These organisms, as you can see, do come in two forms. They can come in a polyp form, which means it sits on the bottom. It's sessile, it sits on the bottom. The medusa form is the free-swimming form. Now, some radiata are sessile only. Some have a mixture of polyp and medusa. It just depends on the particular species. An example of a polyp form would be like a sea anemone. The example of a medusa form would be like your jellyfish swimming around. In between the epidermis and the gastrodermis, there is a non-living layer called the mesoglia. This is kind of like almost think of it as kind of like a glue-like layer, but it's in between. It's hard for me to draw here, but it's in between the epidermis and the gastrodermis, and it's kind of like a proteinaceous acellular layer that holds the two sides together. So the phylum Cnidaria is going to be the major phylum that we'll talk about in radiata. So they do have cells that are differentiated and specialized, much more than what the sponges did. They do have nerve cells and contractile epithelial cells. So if you ever see a jellyfish swimming, it's because they have contractile epithelial cells. They do have enzyme secreting cells that can help them digest food. They have nutrient absorbing cells which can help them to absorb nutrients. They have epidermal cells. They have these super specialized cells that are stinging cells. But they do not have complex organs or organ systems that we've seen in any that we've studied so far. To coordinate their movements and digestion, they do have a nerve net. So this coordinates movements of the tentacles, digestion. It also coordinates expelling waste, capturing prey, and feeding. And so we actually have a nerve net too. If you ever study the human body, you'll find that we have a nerve net in our intestines, spread out all through our intestines, and it's almost like our stomach brain. And if you've ever had a gut feeling, that's part of the nerve net that you have in your stomach region, your abdominal region. So, you know, these cnidarians do have sensory nerve cells and motor nerve cells. The sensory nerve cells allow it to sense and to sense things, but you know, they don't have any deep thoughts or thinking because they don't have a brain. They do have motor nerve cells which will coordinate the movements of those contractile epithelial cells. Their digestion is in a form called extracellular. Extracellular means that enzymes are dumped onto the prey in the gastrovascular cavity. Then the digested food is absorbed into the animal. So that's called extracellular digestion. There is no respiratory system, excretory system, or circulatory system. So nutrients, wastes, gases like oxygen, carbon dioxide, they have to diffuse through the body of the cnidarian. So you don't see any really super thick cnidarians because, you know, diffusion can only work a certain distance from the outside to the inside or from the inside to the outside. So these organisms are typically really quite thin. Another characteristic is that the cnidarians have a mouth surrounded by tentacles, and these tentacles have stinging cells. So if we look up here, this would be where the mouth is, and you can see the tentacles that are surrounding it. If you take a tentacle and you look at it, it's going to have these very specialized cells. Okay, so you can see these very specialized cells right here and right here and right here, and these are called cnidocytes. And when I mean they're specialized, they're really super complex. So they actually have a cell membrane that has what we call a trigger on it. That trigger has the ability to sense the chemicals coming from the prey, and it can detect when it's touched. They also have these really complex organelles called nematocysts. Inside the nematocyst, there's a coiled thread, and there is a microvenom gland, and there is a cap over the surface of that organelle. If you smell the right way and you touch the trigger, the cap will fly off real quickly, water will flood in. So this particular structure is run on hydrostatic pressure. So water will rush in, and that thread will encircle or puncture or impale the prey, and you can see right here it will inject a microdose of venom. So there are barbs and little hairs and spikes that are on these things. So once it grabs a hold of the prey, typically it sticks onto the prey, almost like a fish hook sticks onto a prey. Sometimes these things don't have venom, but they have little lassos, so they'll actually send out a little lasso that will ensnare or entangle the prey item. So they're pretty cool, very advanced complex cells. This is just showing you the touch-sensitive hair-like projection. So you have to smell the right way and touch that. And if you do, the cap flies off, and then the nematocyst will what we call fire. So you'll have little barbs that will stick out and little threads. Sometimes the little threads have microvenom glands. You know, down here deep there will be a little microvenom gland that will send venom into the prey item. And this is just showing you the life cycle of a typical, I guess, a typical Cnidarian. It's hard to say typical because there was a lot of different kinds of Cnidarians, so let's just say that this is the life cycle of one type called Bilateria. And this is showing you the sexual and asexual reproduction cycle. So let's start with sexual reproduction. So little Medusa will be released from the parent animal, and these will swim around. They do have gonads, and they have the ability to produce sperm cells or egg cells. And they do that through the process of meiosis, or reduction division. If sperm cells and egg cells find each other, we will have fertilization. A zygote will form. It'll then go through, a 2-cell stage, 4-cell stage, form a little hollow ball, and will form, in this particular example, a larva. If the larva settles to the bottom, it'll form a polyp, and the polyp will mature. Now that polyp will grow into a colony, so it's kind of like asexual reproduction. Each of these has the ability to produce Medusa, as you can see. And those Medusa, when they come off of the parent animal, they will undergo sexual reproduction. So that's the life cycle of Bilateria. There are different classes of Cnidarians. We have hydrozoans. The hydrozoans are mostly marine, and they include things like the Portuguese man-of-war that you can see in this picture here, and Hydra is a freshwater hydrozoan that we'll talk about in just a second. And the scyphozoans include many of the jellyfish that you're familiar with. All of these are marine, and they have a dominant Medusa stage that swims around. Very reduced polyp stage. So in the hydrozoans, they do have both polyp and Medusa stages, and but some of them are more polyp form and some of them are more in a Medusa form. The anthozoans include things that you're probably familiar with, like the sea anemones, sea fans, and most of your corals. And the polyp is the dominant stage in this particular organism. Many of them are colonial. They form colonies, like a big piece of coral is a colony of animals. And then there's another group called decubozoans, or box jellies. There's not very many species of these, but these are really kind of interesting. They include the things like box jellyfish and sea wasps. So these are ones that are venomous, and we'll talk about those in just a second. They mainly have a dominant Medusa stage. This is just showing you pictorially the cubozoans, scyphozoans, anthozoans, and hydrozoans. So there's your hydra, your freshwater Cnidarian, Portuguese man-of-war. And then anthozoans, you're showing you the coral and the sea anemone. Scyphozoans are your jellyfish there. And then cubozoans would include like your box jellyfish and sea wasp. Now, if you go to some areas of the world, they have very, very, very toxic Cnidarians, and this one happens to be the box jellyfish. And the box jellyfish is highly venomous. It creates a venom that, if injected into you, will go through and it destroys your blood cells. When your blood cells are destroyed in large enough numbers, they leak out an ion called potassium, and if large amounts of potassium leak out of your blood cells, it will actually disrupt the functioning of your heart and muscular and nervous systems, so thus causing heart failure. If you're ever stung by a box jellyfish, in many areas of the world where box jellyfish are common, they will actually have little stations, little aid stations, and all of the lifeguards will have the ability to treat you, but the aid stations will have bottles of vinegar. You can see it's missing in this particular container here, but you'll pour the vinegar on and it deactivates the ability of the stinging cells to keep firing into you, thus minimizing the amount of venom that's injected into you. These are very, very dangerous creatures, and if you go to some places in the world, even in Hawaii, they have warning signs where they'll have the little jellyfish signs warning you that they're there. So let me just kind of go through and show you a couple of examples of scyphozoans. So these are scyphozoans. This one right here happens to be the sea nettle, and we have these in our area, Virginia, North Carolina, and if you're stung by these, that vinegar works pretty well for them as well. So the vinegar will denature the proteins and deactivate the stinging cells from firing. If you do get a tentacle wrapped around you, you don't want to rub sand on it. That would be the worst thing to do. But what you want to do is take a credit card or a driver's license and or a stick and try to pull the tentacle off. The more that you keep the tentacle on you, the more stinging cells will fire into you and inject venom into you. So if you get that tentacle off quickly, it saves you from being stung a lot. So the cannonball is kind of a common jellyfish you'll find, especially in Myrtle Beach at certain times of the year. And then here's just a moon jellyfish, which is commonly seen on the seashore after storms. So these are examples of anthozoans, or this is an example of an anthozoan, and this happens to be a sea anemone. You're probably familiar with the clownfish. The clownfish can swim inside the sea anemone because it has a mucus coat on the outside surface that doesn't allow it to get injected with venom. This is a mutualistic relationship. The clownfish draws in other fish. The sea anemone will inject venom into the fish, eat the fish, and then little scraps or little scraps of fish particles will come out and the clownfish will eat it. The clownfish will poop on the sea anemone, and that feeds the algae. Remember the mutualistic algae living inside the sea anemone? It actually feeds the algae and gives nutrients to that. So that's a mutualistic relationship between the clownfish and the sea anemone. These are just examples of hydrozoans. We've talked about the Portuguese man-of-war. This is actually a colony of animals. There's a different kind of animal that creates this, and there's different animals that create different parts. It's hard to know what to call a Portuguese man-of-war. Is it a superorganism made of many different kinds of animals? It's still kind of up in the air, to my knowledge, as to what that actually is. But it's colonial. It's a colony of animals working together to perform a function. And this is a highly magnified hydrozoan called Hydra. And Hydra is a freshwater Cnidarian. And they're very, very small. They're really super small. That'd be like a super big one there. So this is blown up microscopically. You can see that it's forming an asexual bud right there. So that's a little bud coming off that will pop off and form a new Hydra once it falls off. But they do have sperm cells and egg cells and undergo sexual reproduction as well. Okay, so let's talk about some different kinds of animals. Let's go into Bilateria. Bilateria means that they have bilateral symmetry and they have 3 germ cell layers. Remember, those germ cell layers are going to become, like brain, spinal cord in more advanced animals, middle parts of the animal, muscle tissue, lungs, blood cells, if they have those, and endoderm will become the inner linings of the digestive tract. Okay, so developmentally, these are really important layers of the organism. Not all animals have not all of the bilaterians have really complex structures, but many of them later on you'll see will have really complex structures that will come from these tissues. All right, so Bilateria is divided into protostomia and deuterostomia, and it all depends on what is in the first hole that forms in the young embryo. What is the fate of that particular hole? So in protostomes, the mouth forms first, and in deuterostomes, the anus forms first. Now, this particular group, Bilateria, is going to have a series of different kinds of fluid-filled cavities. Some of them are going to have a true coelom, some of them are going to have a pseudocoelom, and some of them will not have a coelom at all. But these fluid-filled spaces between your organs, your body organs, and the wall of the body, this fluid-filled space is called a coelom. And the coelom is a relatively important structure because it does various kinds of things for the creature. For example, it provides shock absorption. Okay, so in between your body wall, your abdominal wall, and your digestive tract, you have a coelom that your digestive organs actually sit in, and it's fluid filled. And every time you run or jog, you know your organs aren't being slammed up against the wall because there's a fluid filled shock absorbing cavity there. The coelom also provides a space for organ growth and development, so it's a space that organs can actually grow into and grow from. The coelom does provide for flexibility of the animal. So in many animals it forms a hydrostatic skeleton. So water pressure is pumped in there, and that gives them a skeleton, a structure. All right. So there are different kinds of coeloms. There are no coeloms. We call organisms with no coeloms a coelomate. An example will be flatworms, and we'll study some flatworms in just a few minutes. We then have eucoelomates, which do have true coeloms, and these are some of the organisms that do. So segmented worms, mollusks, arthropods like insects, echinoderms, which are starfish, and chordates like you, we have true coeloms. And that means that inside of the mesoderm, the coelom actually sits inside the mesoderm, and it's surrounded completely by the mesoderm. And that's where many of our complex organs, like lungs and digestive organs, actually sit inside of that coelom. And then there are creatures that are pseudocoelomates, and these would be the roundworms that we'll study. And here you can see the pseudocoelom exists in between the endoderm and the ectoderm, excuse me, and the mesoderm. There the pseudocoelom sits. It's just a space that's in between those particular layers. Okay, so that's the difference. You know, you have to be inside of the mesoderm, in the middle of the mesoderm, to be a true coelomate, but outside of the mesoderm and in between the endoderm to be a pseudocoelomate. Just showing you one more time the comparison between protostomes and deuterostomes. So if you look at the first hole that forms in the little embryo that's forming, if it becomes a mouth, you're a protostome. If it becomes an anus, you're a deuterostome. Just showing you, I guess, one last picture of that. So the blastopore, if it becomes a mouth, you're a protostome. If the blastopore becomes an anus, you're going to be a deuterostome. So let's talk about the protostomes for just a few minutes. Protostomes are divided into Leucotrophozoa. I know it's a huge, weird word. It describes the larval state or structure on a larva of those animals. And a Metazoa means shedding animal. But really, molecular data, that is, you know, the sequences of DNA and RNA, are going to be used to differentiate these particular groups. So the Leucotrophozoans include the phylum Platyhelminthes, again a weird word, which means flatworm, Rotifera, and Mollusca, and Annelida. So these will be the phyla of Leucotrophozoans that we'll study. And we'll start with the Platyhelminthes, the flatworms, first. These particular organisms can be free-living, which means they move around freely. They don't have to parasitize things, but others of them are truly parasitic. They have to live on the tissues of other living organisms. Reproductively, they can be monoecious, okay, which means they have both male and female parts at the same time, but they can also reproduce asexually through forming buds and breaking pieces of their bodies off. Their digestion, they have a gastrovascular cavity, much like Cnidarians, so they have a mouth anus. They don't have a separate mouth and anus like you. So these organisms, because they're bilaterally symmetrical, so I'm just going to draw a little planarian over here, a little free-living flatworm. Because they have bilateral symmetry, we begin to see cephalization, which means the formation of a head end and a tail end. They do have primitive sense organs located in the head end. So there's a little eye spot right there. And they will have little collections of brain cells called ganglia in the head region. So we begin to see cephalization that we didn't see in the other organisms that we saw previously. They do have a unique excretory system. So this is the first time we've seen an excretory system. The excretory system's job is to get rid of liquid waste products out of the body of the organism. And they have these little specialized things called flame cells that we'll see. And they do not have a circulatory system or a respiratory system. So again, this limits the thickness of their body because the gases are going to have to diffuse through the skin of the organism and to get oxygen in and carbon dioxide out. So they can't be super thick. There's no super thick platyhelminthes that you'll find. And these are acoelomates, so they don't have a coelom or body cavity that their organs sit in. The main groups or classes, remember we're in the kingdom Animalia, phylum Platyhelminthes. These are the classes over here. We have Turbellaria, which are the free-living flatworms. An example would be Planaria. So we'll see planarians in 1 minute. We then have the Cestoidea and Trematoda, and these are going to be the things like flukes, like liver flukes, and tapeworms. These are parasites, and we'll look at those, and we'll not really look at the monogenes because it's something you probably will never see, but those are parasites a lot of times of freshwater or marine organisms. So an example of a tubularian worm would be Planaria. So this is Planaria. This is a really common organism. They've done lots of studies on planarians, and they're really small. That would be a big one. I'm just making a little dot on the screen. That's like a super big one. So they you can see them with the unaided eye, but they're typically relatively small organisms. Again, super thin. You can see there's ganglia. There is collections of nerve cells, and it's kind of like a little simple brain. There are eye spots that can sense light and dark. They do have nerve cords that extend down so they can have sensory information given to their ganglia through different parts of their body. And they have a gastrovascular cavity. Now this is a very specialized gastrovascular cavity. If you notice, it's highly branched. So everywhere there are body parts, there's part of the gastrovascular cavity. It's branched all throughout the body. So the reason that they have a gastrovascular cavity that's branched out is because they don't have a circulatory system. So they can't circulate nutrients. They can only have the gastrovascular cavity go all throughout the body so nutrients can be given to all the body cells. So everywhere you have cells that need nourishment, you have to have parts of the gastrovascular cavity. There's that mouth anus, and that's right there. And it does form a little tube called a pharynx. That tube is almost like a vacuum cleaner. It'll come out and suck up particles or nutrients, and it can be extended and then retracted back in. All right, well, there is a problem in this animal. How do you get rid of liquid waste products? And they've solved this by having a very primitive excretory system. It's made up of the flame cell, okay, or the flame bulb. So this will be a tubule right here that connects many flame bulbs together. All of the waste products that they form are going to be excreted out of the body of the animal through the nephridiopore. And so the flame cell will take interstitial fluids, so any of the water, aqueous water, or fluids in the body called interstitial fluid, they'll create through these little cilia that are right here. These cilia will move in a motion that will bring the interstitial fluids into the actual flame bulb, and these any waste products can actually be filtered out and taken down and dumped out of the organism. Okay, so it's kind of almost like a primitive kidney of sorts. But if you go to the internet and type in flame cell, you'll actually see they flicker because they move so quickly. It looks like a flickering flame that you would see on a candle, and it's kind of why they call it a flame cell. These platyhelminthes are beautiful. If you look, all of these are marine flatworms, and they come in all different shapes and colors and sizes, and they swim, and some of them are really big, and they're just kind of really strikingly beautiful creatures. Okay, so some of the more interesting ones will be the trematodes, the flukes. You can see this is the egg of a fluke, and this is the adult form. And inside of it, you can see a blood meal. So it's been eating blood. These can be dangerous to humans because they are blood eaters. The flukes have some really complex life cycles. So let's start with an infected human. The infected human will basically excrete these out through feces. If they get into water, if the little embryos get into water, they'll actually live part of their life cycle inside of snails. They'll then hatch out of snails and swim and burrow into the muscle tissue of fish. And then if humans eat raw fish, then they'll begin to grow inside of the human. Inside of the human, they'll actually bore through the intestinal wall, and they'll move to places like the liver or the lungs. So the lungs would be over here. So they'll move and live inside as an adult inside the lungs or the liver or the urinary bladder, and they'll release eggs into the lungs, into the digestive tract or into the urinary tract, and then people will excrete these things out. So these can be very dangerous because they drink blood from you. So the way you stop the cycle of infection is you stop eating raw fish. Many countries actually kill snails. And of course, we treat waste products in a sanitary fashion so that we don't keep spreading these particular organisms. So there are ways that we can stop the cycle of infection. Cestoidea includes your tapeworms. Aren't they cool looking worms? Here's one under scanning electron microscopy, and you can see that it has all of these little hooks on the head. And it actually has suction cups on the head too. This is how it adheres to the wall of your intestine, so that when you have peristalsis or waves of movement inside your intestines to get rid of feces, they can actually hook on or suck onto the wall of your intestine. This picture down here would be an example of a microscopic view of it with the little hooks and the suction cups that you can see. They grow pretty long. I think they found tapeworms as long as 30 feet inside of people, and they're really interesting creatures because they don't have a digestive system. They absorb your food that you eat, so you digest the food for them, and they absorb it right across the wall of their body. And it's an egg-laying machine, these things called tapeworms. So if you notice, they have these little sections right here called proglottids. These proglottids are little reproductive factories. Okay, and they just keep on reproducing. If you ever have animals with tapeworms and you look at their waste products, you can see the little white parts inside of their feces. So take a look next time you change the litter box of your cat, or if you have a dog, pick through its poop and see if they have little proglottids there. So in the life cycle, humans will poop these out. The eggs will get onto grass. Animals such as pigs, cows, and there are fish tapeworms too, will eat the eggs, and they'll actually live inside of their muscle tissue. Inside the muscle tissue, they exist as a little bladder worm. And when humans eat undercooked pork, beef, or fish, they'll actually hatch out and they live in the intestines. So they'll live in the intestines of humans. And they become little egg factories, so all the eggs will come out in the poop of a human. The way that you test to see if you are infected with tapeworms is they'll take a poop sample from you and look at it under the microscope, and they'll see if there are eggs there. So rotiferans are a different group. I'm not going to spend too much time with these because you probably will never see one because they are typically in pond water and they're small. But if you ever look at the head of the organism, they have these really complex heads with cilia. Each one of those little lines there is cilia. And these cilia create currents, water currents, where they helps them to actually feed. Their three main body parts include the head, which contains a bilobed brain. They do have eye spots, and they have a corona, or a a circular portion of cilia that surrounds their mouth. They do have a trunk which has all of the digestive organs, and then they have a foot that can secrete a sticky material that helps them to adhere to the surface of objects that they're where they live. They do have a complete digestive tract, so they do have a mouth and a separate anus, and they are filter feeders. They are free swimming or planktonic, and they do have separate male and female, so they're dioecious. And they are pseudocoelomates, so they have a pseudocoelom, not a true coelom. And this is what they look like. So you can see the mouth and the anus, so they have a complete digestive tract where food comes in one way and poop goes out the other. So their mouth leads into to a pharynx, and they have a mastix, which is an organ that chews the food up. They have digestive glands that add enzymes to the material. They do have a stomach and an intestine. Stomach helps digest food. The intestine helps absorb and digest food. And then they have an anus where the waste products go out. Here you can see the pseudocoelom, a little space right there. And they do have ganglia, a bilobed little brain-like structure. And then there's their corona of cilia that you can see surrounding its mouth. I'll just quickly show you a little picture of a rotifera, a little video of a rotifera. So you can see it's moving around. It's stuck to something, but it can actually detach. And it has a little head that has there we go, it's moving around now. So you can see the little cilia moving around, and you can see the mastix pushing food into and crushing food up and pushing it into the stomach. So it's a filter feeder. And you can look at a longer video of this if you'd like to. I'll put it on Blackboard for you to look at. And I think I'll stop right there, and we'll do a part 2 of Introduction to Animal, and we'll start with the mollusks on the second video.